Our next speaker is Dr. Helen Vuong. She's currently a postdoc at UCLA studying the role of the maternal microbiome in fetal brain development. Dr. Vuong earned her BS in neuroscience from the University of California, Los Angeles, UCLA, and she continued her graduate education at UCLA, earning her PhD in molecular, cellular, and integrative physiology under the mentorship of Dr. Nicholas Brecha. Without further ado, everyone, please welcome Dr. Helen Vuong as she dives into the importance of the microbiome in development.
Hi, everyone. Thank you for good morning, first of all. And I appreciate you guys spending your Saturday morning with me. I'm going to try and figure out how to share screen. I'm still getting used to this whole Zoom meeting. Let's see. Oops. All right. Thank you, Diana, for organizing this STEM Enrichment Youth Conference. It's amazing that you're able to put this together. I saw in the chat that it just started this year and you just started in June or something like that. So just in a few months, that's amazing. And so I am very excited to be able to share with you guys some of the work that I've been doing and what I can impart on you at this stage of my career. And so just as a title, most of it's going to be focused on the gut microbiome, brain and behavior and health and disease, but I kind of want to inject some previous work that I've been working on as a graduate student. Okay. So like Diana was just saying, I did my undergrad at UCLA. I did it in neuroscience. I didn't actually think that I was going to be doing neuroscience. I really did enjoy science in high school, but I didn't know what I wanted to do once I got into college. So if you're still figuring it out now, that is, I think, absolutely okay. I know lots of people that change majors in the middle of their college and figured out, what they wanted to do later on. I also did my graduate career at UCLA in MCIP, and that was with Dr. Nick Brecha, and we did, I'll explain that a little bit later about retinal visual processing. And then finally, I did my, I'm doing my postdoc right now at UCLA with Dr. Oops, sorry, what's going on here? Okay, sorry. With Elaine Shao at UCLA, and that's where the bulk of my work is right now, is looking at microbiome on host behavior in her lab. And in the middle of all of this, I also had a son, and here his cute face, and very proud and happy to have a healthy baby boy during these COVID times. So that's what you can look forward to if you would like to have children in the future. And so just to go over really briefly, some of my graduate work was looking at neural cells in the back of the eye. And these are called retinal cells, and they process visual information before it goes to the brain. So all that light and shadow and visual processing begins in the retina. And my work really focused in on how these amacrine cells communicated with use a laser pointer, communicated with these retinal ganglion cells. And I used different techniques, including looking at morphology, using confocal microscopy. I looked at how two different cell types communicated with each other, formed these contacts, and I also looked at their functional output. So taking an electrode, sticking it into these cells, and then recording the electrical activity. And you can see these little spikes. These little spikes indicate that there's some firing and communication happening. And if you take a drug or you stop the communication, you lose those spikes from happening. And that's why you get these decreased spikes. And so most of my graduate work was really looking at this model of neural communication in the retina. And when I finished my graduate work, I was very interested in understanding whether our body was specifically isolated in how it functioned. And it turns out we're not isolated. What makes us us is actually very interesting. We co-evolved with trillions of microorganisms. These include bacteria, virus, fungi, and that these microorganisms inhabit our body, and they've co-evolved with us, and they are in our body and on our body, and in different locations of our body, they're all very unique in terms of what are the microorganisms that are present, and they're have a very variable composition. Just by looking at these micrographs that are about 3,000 fold magnification, you can see that these little microorganisms that aren't really visible to the human eye are very different from location to location. And so we have only really scratched the surface in this field about understanding what each of these little bugs are doing and how they could be potentially influencing our host physiology. And so one of the kind of cool projects that have come out about the microbiome is the Human Microbiome Project, which was a National Institutes of Health initiative that started around 2007 to 2008. And what they essentially are trying to do in this Human Microbiome Project is to profile all the different types of microbiome in healthy people, as well as people that have a disease model, like IBD or diabetes, or even women that are pregnant. They all want to this project essentially wants to do is profile all the different microbiomes. And some kind of cool facts that came out of this project is that microbes, in terms of their numbers of cells, actually outnumber the cells that make us us, from 1.3 to 1. And if we look at the genetic material from microbes versus human genetic material, they actually outnumber us 150 times. And if we think about all the different types of microbial species that are present, there are over 10,000 different ones on our bodies. And what is really interesting is that a lot of it is accumulated in our GI tract, and 95% of the microbiota is in our gut. And if you take all that microbial mass from our body and put it into a container and try to measure its weight, it's around 2 to 6 pounds. And just to give you some perspective about what that means, our brain is about 2.6 pounds. Our heart is about 0.7 pounds. So it's very interesting that something that we never really thought was us is actually either the same or more than many of our other organs in terms of weight. And so based off of this information, I became really interested in how these little bugs could essentially be affecting our body. And research has been ramping up, and scientists have kind of gone into these different avenues and try to understand what the microbiota is doing in terms of affecting metabolism, how we process food, or the immune system, how we can fight off different types of infections, drug metabolism. So how quickly or how efficacious our drugs are being used in our body is dependent on the microbiota. Intestinal function. So if someone has ever had GI disorders, intestinal motility issues, this could be due to the imbalance in the microbiota. Bone homeostasis, development of the bone has been implicated in microbiota. And then what I'll really focus on today is how the microbiota is affecting our brain and behavior. And so this idea of microorganisms affecting host behavior is not new. It's actually been studied in many different organisms. And so this Ophiocordyceps unilaterus is a fungi. And essentially what it does is it hijacks the ant's body to actually cause the ant to climb to different levels of a plant so that it could get more or less sunlight to grow. Another example is the rabies virus. It can attack, most commonly known in dogs, and really causes aggressive behavior. And the idea of causing aggressive behavior is to actually cause these dogs to bite other dogs and spread the rabies virus. So again, it's to promote their development or their proliferation. We have Toxoplasma gondii. I'm not sure if people have heard of this, but it's more commonly known as toxo. What scientists have found is that this particular Toxoplasma gondii is able to infect mice. And what they do is it causes the mice to lose aversions to the cat, and it will lose aversion to cat pee, for example, and it'll cause the mouse to actually go towards the cat. The cat will eat the mouse, which will allow for this Toxoplasma gondii to proliferate, because it only really survives or can live in the feline gut. And then finally, there's the Salmonella enterica. It also affects our feeding behavior, and ultimately the goal is to proliferate inside the human gut. And so my work is going to be focused on not necessarily all those other microorganisms, but the gut microbiome in particular and how it can affect host behavior. So this is just a pictograph of a intestine that's been stained with different markers that are localized to either the mucosa, some intestinal cells, and then this is the lumen over here, and all these different colors over here are different microbes or bacteria that are present. Oh, sorry, am I drawing? It's weird. Okay, so, and then the idea is that we are interested in how those bacterial products or these different bacteria could be communicating with the brain. And scientists have begun parsing the role of direct communication through the vagus nerve. So there's this particular nerve that goes from the intestine all the way up to the brainstem. There's also the idea that there are neurotransmitters, hormones, or small molecules that come from these microbes or the host that go through the circulatory system and communicate with the brain. And then finally, there's the idea of there's this communication between these microbes and the intestinal immune system that could also be creating MAMPs or metabolites that communicates with the brain. And so we are actually really most interested in the circulatory system pathway, but there are tons of research in all of the three different linking pathways. And how do we study this? It's actually really hard to do it in humans because you can't really isolate specific bugs and then take out the brain of a human and then do all these behavior stuff on them. So a lot of the work that we do is using animals, and we use a technique called neurobiologics, which is it means known life. And what we do is we use mice and we put them in these isolators or these bubbles. And in these bubbles, they are completely sterile. They lack all types of microbes. And so this allows us to really control for what kinds of bacteria or whatever microorganisms we're studying and look at what their effects are. And so essentially what we have are we take these mice and we can either make them germ-free, which is in these isolators. Alternatively, we have this technique using antibiotics, which will clear all or most of the bacteria in the gut. We also have specific pathogen-free group, which is essentially our control group. They have the full microbiome, nothing's been done to them. They're completely normal in the sense that they have a full microbiota. And then finally, we have the specific bacteria. So we colonize these mice with specific bacteria that we're interested in and trying to understand what their role could be. And so previous research from scientists have already begun understanding what the role of the microbiota is doing in animal brains. And so, for example, in germ-free mice, there are decreased expression of these key brain genes like NGF and BDNF. As you can see, comparing a control animal with a germ-free animal, you see there's a significant decrease in the amount of bright spots. Similarly, there's an altered stress response in these germ-free mice. So usually when animals are stressed, they release these different types of molecules, but then in the germ-free mice, they have this altered level of these molecules. There's also anatomical changes. Germ-free mice have these different dendrite processes and microglia formation. And also there is a altered blood-brain barrier integrity. So there's tends to be a more a leaky brain where other molecules can easily enter the brain in these germ-free mice. And not only is there these sort of anatomical changes, but there's also been behavioral evidence suggesting that the microbiota has a major influence on how animals will act. And so some tests that scientists can use is this social interaction test where they put a mice into a 3-chamber plexiglass box. They put them in the center, and then they will either time how long long they spent in the chamber with the mouse that is a novel mouse or an inanimate object, or how many times they enter one versus the other. And typically, mice or rats are very social animals, so they'll tend to go towards the object or the other mouse. And what they found was, yes, in a control animal, they spent a lot of time with the novel animal. But then if you deplete the animal of all of its microbes, it becomes less social. It actually spends more time with the inanimate side versus with the animal. But if you take that germ-free animal and you gave it back some bacteria, it actually goes normalizes itself. It comes back and interacts with the novel animal. So it's very interesting how these bacteria are now able to influence different behaviors. And then here's another example of another behavior, which is an open field test. You put the mouse in the center and you see how much time it spends in the center versus on the outside. And typically, rodents like to spend their time on the outside versus in the center, because center typically means danger. And so interestingly, the control spends a little bit of time in the center, but then the germ-free one spends a lot of time in the center. So they have this change where they are not as scared. And so they spend a lot more time in the center compared to the outside. And here's just some recent evidence suggesting that the microbiome is not only affecting baseline behavior or baseline anatomy, but it also has a very interesting role in modulating disease phenotype, in particular neurological diseases such as Parkinson's. And in this paper, they look at germ-free animals in an alpha-synuclein Parkinson mouse model. And they show that this animal is able to, it has an altered Parkinson's response or time to cross when you deplete the microbiome. Similarly, there's another paper looking at ALS, and they do a bunch of behavioral readouts in the animals with the ALS transgenic mouse line. They actually have an altered response when you deplete the microbiome. And so this is all really interesting in highlighting what we want to do in our research group. And that is, again, focused on brain function and behavior. We wanted to take a step back and we wanted to understand how the microbiota impacts brain development. So as an infant or a prenatal animal, how is the microbiota affecting brain development? And then what is the role of the microbiota in seizure development in a disease model of seizure? And so to jump in on the microbiota side of how it affects maternal development or offspring development from the maternal environment, previous work by Elaine Shao, she looked at how different autistic-like behavior, such as stereotype behavior using marble burying, when the moms were exposed to an infection, which is a mouse model of autism, the offsprings like to bury the marbles a lot more. But if you gave the mom back this Bacteroides fragilis, it actually reduced the amount of burying. Similarly, the amount of communication is different. So in the mouse model of where they have the maternal immune activation, they have the offsprings have reduced number of calls, the call times are reduced, but then if you give them back this Bacteroides fragilis, they have these normalized communication in these offspring. And so our work is focused in on what about the maternal microbiota is affecting the fetus. And so we asked some key questions. What does the physiological maternal gut do for a brain? And what is the mechanism? And so what we did was look at the gene expression. Essentially, this is just a heat map looking at up or down-regulated genes. And we found a lot of these key genes were related to axon development. And so when we take embryonic brains from mice that, or their moms had a complete microbiota or a depleted microbiota, we saw that the fetuses had reduced axons compared to in the maternal microbiota that's depleted compared to a full microbiome. And not only do we see brain differences, we saw behavioral differences. And some of the behaviors we saw were these tactile sensory behavior. These microbiota depleted offsprings have a longer time to respond to these stimuli. And this suggests that they have this sort of hyposensitivity in their peripheral response to sensory stimuli. And just as kind of this is a random forest analysis using machine learning, which is kind of something that Akshaya touched upon. Essentially what they did was they looked at these small molecules in the maternal microbiota that's produced from these microbes and try to see whether or not these little small molecules could affect brain development. And so taking these small molecules and pairing them with different microbiotas, we're able to predict with 87.5% accuracy that these bugs, or sorry, these little metabolites could be associated with a particular maternal microbiome. And what's really cool is that we found some that were decreased in the microbiota depleted animals compared to the non-microbiota depleted animals. And that if we gave those small molecules back into the mom and looked at the embryo brains, we can see these axons grow right back out. And not only do we see the sort of anatomical changes, but we see the behavioral restorations where they have normalized tactile sensory behavior. And so this is just in summary that the maternal microbiome produces these small molecules that affect growth of these axons. They get to the brain, and then it also affects behavior. Let me just check how we're doing on time. I think I have a few more minutes here. And just to kind of touch on the second story, which is how we looked at epilepsy. So epilepsy is a neurological condition based off of disturbances in electrical activity. And the refractory seizures that occur in children are really uncontrollable, and they don't really respond to anti-seizure medication. And it's a worldwide burden, with 65 million people around the world having epilepsy, and 1 out of 3 have uncontrollable seizures that don't respond to treatment. A kind of not so new treatment, but has its downsides, is the ketogenic diet, which is a high-fat, low-carb diet. It's been used to treat infantile spasms, Dravet syndrome, GLUT1 deficiency, and people have used it, or scientists have used it for infants using a gastronomy tube. However, the ketogenic diet, if anyone has ever tried the ketogenic diet, it's just basically Crisco. It's very harsh. It doesn't taste good. I mean, you can imagine as a child trying to take ketogenic diet every day is a terrible way of going. And so this kind of motivated us to kind of understand what the ketogenic diet is actually doing. Could it be doing this through modifying the microbiota? And then could we then harness this to develop something that could be more efficacious in treating childhood epilepsy without using the diet? And so we used a mouse model of 6 Hertz psychomotor seizure in collaboration with a graduate student in our lab, Christine Olson. And what we show is that the ketogenic diet is effectively reducing the seizure threshold. So it takes a higher stimulus to induce a seizure when you are taking the ketogenic diet. The ketogenic diet significantly changes the microbiome. You see, this is the starting microbiome over here, and then through the different diets, a ketogenic diet versus a controlled diet, you have very different microbiomes. And what we found was that 2 particular bacteria are sufficient for carrying out this ketogenic diet effect. And one of those is Akkermansia acidophila, and the second one is Parabacteroides. If we took those 2 bacteria and colonized the animals with them and then did the seizure testing, we saw that these bacteria effectively colonized the gut, and that it also increases their seizure threshold, which is really cool. We were able to find that they don't work independently of each other. So if you just gave them Parabacteroides or just Akkermansia, it doesn't work. You have to give them 2 together. And if you gave them a random bacteria like a Bacteroides, it's not as effective. We used finally another model, which is a KCNA, which is an actually occurring transgenic model of seizures. And we get these really, you do EEG EEGs, which is electrical activity in the brain. We record that. And what we found is that in these animals, they typically have these really crazy seizures. They spontaneously occur. And you can see here that there's a lot of uncontrolled brain activity that causes them to jump around. When we give these KCNA mice back some of this Akkermansia parabacteroides, we're able to reduce the seizures and the time that they have seizures. So this is really exciting for us that we were able to find something so effective. And we wanted to figure out how it was doing this. And so just very briefly, we again looked at small molecules that were produced in either the blood, or it was produced in the gut that makes its ways into the blood or is still in the gut. And then we looked at the actual GABA to glutamate ratio in the animals. And we found that Akkermansia parabacteroides actually normalizes this GABA to glutamate ratio, allowing for normal brain activity. And just in brief, this ketogenic diet regulates the gut. It regulates these specific metabolites in both the colon and in the blood, and essentially increases this GABA glutamate ratio to cause seizure protection. And so hopefully you guys are interested and amazed as much as I am about this gut microbiome and how it could affect not only the developing brain, but a diseased brain. And our goal is to continue to understand how we can harness this gut microbiome to treat health and disease phenotypes. And I know I'm short on time, but I just want to briefly say that in addition to doing research, I'm also an educator. After you finish your grad school and you go into doing a postdoc, there's a lot of different avenues for you to pursue. It could be going into industry, it could be going into academia, it could be going into science writing. There's all different types of ways you can take your PhD. And in my case, I want to go into a faculty position where I get to continue doing amazing research, but also teaching. And so one of the programs that I'm a part of is called Uplift. It's an NIH-funded transition award for postdocs to be able to teach. And part of my teaching responsibilities happens at a sister institution at California State University, Los Angeles, or CSULA. And these are some of my mentors as well as my colleagues, and we spend a lot of time driving back and forth in LA traffic. Well, we did, not so much anymore, but to teach at CSULA as well as do research. And so teaching is a great passion of mine. And if teaching is something that you find interesting, then doing academia is a great way to do it. Or you could be teaching without research. There's also that too. And so I just also wanted to thank everyone that's in my lab, my mentor, obviously, Elaine, all of the PhD, postdoctoral, and of course, all the undergraduates that have been instrumental in making this research project possible. And so with that said, I will take any questions.
We're a little short on time, so if you guys have any questions, just ask as soon as you can.
Sorry, let me see if I can read. How long did the research take? Oh, the research took many years. Different ones, different projects take much longer. And so at least for the maternal microbiome one, that one took around 4 years and a year in review in the journal before it got accepted. And yeah, it varies. It depends on how big your question is and how thorough you want to ask it. So I would say in most parts, it takes years. And then do you take high school student researchers? We have taken high school student researchers. It's some of them came through programs, some of them came through recommendations. If you're interested in doing research, I would definitely look through the different websites, find a researcher that you're interested in, and just I would email them. I wouldn't cold like show up in their office, but definitely go email them and take the initiative to reach out and see if you can get a research opportunity. I think we are, at least for most of us, having undergraduates or high schoolers is a great way to get started on research, especially during the summertime too.
Any last questions? Anybody? No last thoughts?
Yeah, and I'm sorry I didn't put up my email, but if anyone has any other further questions, you can email me. I don't know, I guess I could reach out to Diana and then she can forward that information to everyone.
Yes, definitely. I can send it through the Slack.
Okay.
Okay, awesome.
Thank you, everyone.
Yeah, thank you so much, Dr. Vuong. I know it's very early and you know, you're very busy, so thank you so much. I hope everyone really enjoyed learning about Dr. Vuong's journey through microbiome research.